Okay, oops. So the last, there we go. Last lecture for this chapter is about oncogenes and tumor suppressors. So these are genes that are naturally encoded in your genome. And we've talked about how your cells are assaulted with DNA damage all the time. Outside sources, inside sources, issues with DNA replication. So, and we have many DNA repair mechanisms that can fix a lot of these mutations. But your cells have mutations in their genome now. And which mutations really matter when we think about cancer? And it's really mutations in these two different classes of genes. So oncogenes, and we'll also use the term, oops, that doesn't fit, doesn't like me writing up there, proto-oncogenes, okay? These are like the gas pedal. So let me back up. Both of these types of genes primarily regulate cell cycle in one way or another, directly or indirectly. Oncogenes normally tell the cell, yeah, it's time to divide, grow, make more. And tumor suppressors normally say, hey, wait, let's make sure everything is working okay. Let's make sure the DNA is repaired. Let's make sure at the checkpoints the cell's ready to go through cell cycle. So these are proteins that naturally work in your cells to regulate cell division. Okay. So you have your normal go and your normal stop, right? And just like on a car, you need to stop and then you need to have gas to restart, go. So they're both important. And so on their own, they cause no issues. They're normal cells. It's when they are mutated or they aren't expressed correctly that causes problems. So, proto-oncogenes. You have this table in your textbook, and I would suggest you have it out for the quiz and the Unit 4 exam, just as a reference. No point memorizing these, and I can't even remember if RAS is an oncogene or a tumor suppressor the semester after I teach this class. It's just, that's, I don't think, the important part. The important part is to understand a little bit about how these cells, or sorry, how these proteins work. And if you're reading through the book, we're not going to focus on all the different ways they can get mutated. We're just going to look at what their functions are. So the reason we call them proto-oncogenes is because they normally have these positive roles in the cell to promote cell division. When they're mutated, we then call them an oncogene. Okay, so oncology is the study of cancer. So these are cancer-promoting proteins when they're not being regulated right. And really what's happening is, in general, you either have a mutation in this protein that can basically cause the protein to kind of work on its own. So to function without outside regulation. Okay. Or, and I'll give you some examples of these in a minute, you can have a mutation in which the protein is overexpressed. And overexpression means there is much more than the cell normally has or needs to function. Excuse me. And with either one of these types of mutations, the outcome is continuous stimulation of cell division. Oops. Sorry. Of cell division. And or the inhibition of apoptosis, which we talked about, that's an issue, right? Because if a cell doesn't undergo apoptosis, then it's not going to die off, and you're going to have this excess cell buildup. Okay, so let's just talk a little bit about these different classes of oncogenes. So I'm going to see if I can do this. Okay. So if you look at this table, okay, you can have proteins that are growth factors that can become oncogenes. And what's happening is, and remember that different cancers have different characteristics. So cancers don't have all of these. Some cancers have some cancers have others, some have a combination. Some cancer cells can produce their own growth factors. I'm going to put GF, growth factors. Right? And so if a cell is producing its own growth factor and releasing that and has a receptor that it binds to, and then a signal is transducted to divide, remember this is called autocrine signaling, telling yourself what to do. And so some cancer cells can stimulate themselves to keep dividing. Okay? So they might produce their own growth factor, or they might produce excessive growth factor. So they might also start telling the other cells around them to reproduce. Okay. Let's look at the second class. Receptors. Sometimes these receptors are mutated, right? And the receptors are these little guys on the outside of the cell. They're mutated so that they don't have to bind a ligand in order to turn on a cell proliferation pathway. Mutated so they don't have to bind a ligand to stimulate cell division. Okay, so instead of having to bind epidermal growth factor, the epidermal growth factor receptor, this oncogene, now can just keep telling the cell, hey, let's divide. Don't care that there's not an outside signal there. Let's just keep dividing. Sorry. This is called ligand independent signaling, because the cell signal is happening independent of the ligand. And this is also called a constitutively darn it, I don't know why it does that to me. Active receptor. That means it's always on. So it's not waiting for the signal to tell it to be on, it's always on. Okay, so you can have cancer cells producing their own growth factors or excessive growth factors, or the receptors that bind to these growth factors work without even having to bind to the growth factors. Let me erase this. This third class, and RAS, is one of the most common mutations in cancer. you'll see RAS and P53, a tumor suppressor. What happens when these GTP binding proteins are mutated, like RAS, is they stay bound to GTP, which means GTP is not hydrolyzed to GDP. So if you remember G protein or GTP binding proteins or G proteins, right? They're not active until they're bound to GTP. Then they're active for a while. And then an RGS comes and hydrolyzes or stimulates the hydrolysis of GTP to GDP. Now the RAS or the G binding protein is no longer active. Bonding nucleotide exchange factors come on, give it a GTP, and it goes through that cycle. So with this mutation, the GTP does not get hydrolyzed, so it's always active, the G protein. Okay, so if you look back through some of those signaling pathways, the signal is constitutively or always being turned on due to a mutation in something like RAS. Class 4 is similar. Remember, kinases means they phosphorylate other proteins. And we've talked about how that is important in cell signaling pathways. And so when these kinases are mutated, they're continuously phosphorylating other proteins. So they're continuously stimulating pathways. So you could go through and look what would happen if MAP kinase was always on. Okay. Transcription factors. Remember that transcription is DNA to RNA to make that mRNA. Okay. And so, let's see. Sorry. Try to give myself a little more room here. If transcription factors are mutated, they're either hyperactive, so they're stimulating gene expression more than they should, or they're produced at excessive levels, which both of them are going to increase the levels of messenger RNA, which is going to increase the levels of proteins made. So mutated transcription factors, oncogenes, are again stimulating cell division by turning on genes at levels they shouldn't be on. And then this last cell cycle or apoptosis regulators, okay, the cell cycle ones, you recognize, cyclin and CDK, right? And you remember these are directly stimulating cell cycle. So when you have these mutated, you have a loss of the checkpoints control, loss of checkpoint, let me just write it out, checkpoint control for cell cycle. Right, so if you remember way back here, we talked about these different checkpoints to make sure the cell is healthy and ready to divide. And if you look back to that chapter 24, so CDK and cyclins went up and down in levels depending on when they were needed to activate cell cycle. So if you have mutations in them, then they're going to continuously, they might not get broken down, and continuously stimulate the cell cycle. Okay, and if you have an apoptosis regulator that's mutated, like BCL2 or MDM, and remember MDM is in the P53 pathway, then what's happening is you're not allowing apoptosis to happen, which means the cell is going to survive, even if there's things like DNA damage or something else that's telling the cell, you really need to self-destruct, you're no good anymore. Mutations in these type of proteins are not going to be able to stop, or are not going to, why do I keep getting messed up when I say this? Are not going to work, and apoptosis is not going to happen. The cell is going to survive. Let's just leave it at that. All right. Your other big group of proteins are called tumor suppressors. These normally stop cell cycle, just like the brakes on your car. The mutations in them are called loss of function, which means they can no longer stop the cell cycle. Okay. There's two big classes of tumor suppressors. The gatekeeper genes are involved in controlling cell proliferation, so cell division. Again, P53 is one of the most frequently mutated genes in human cancers, because remember, P53 stops cell cycle in response to DNA damage. Well, if it's mutated, you can still have DNA damage, and the cell is going to go through cell division and produce more cells with that same DNA damage. The other type of tumor suppressors are ones that are involved in DNA repair. And we didn't talk about the mechanisms of DNA repair. Again, you should get that in genetics. But when these are mutated, you get genetic instability, which means now your genome is even more susceptible to mutations. And one of the caretaker genes you're probably familiar with is BCRA1, BCRA2, the breast cancer genes. So these genes are normally involved in double-stranded DNA break repair. So if your DNA gets broken, both strands, BRCA1, BRCA2 come in and help with that repair mechanism. So if they're mutated, you have a default in repairing those mutations correctly, and you're going to gain more mutations, and that's what's genetic instability. So the last two slides we're going to talk about are the two-hit hypothesis and genetic instability. So some people talk about cancer being inherited, okay? And you don't really inherit cancer, but you may inherit one gene. This could be RB, this could be BRCA1. You may inherit one that's already mutated. Right, and you remember from genetics that you have two chromosomes, so duplicate information. You have two BRCA1 alleles, one from mom, one from dad, and maybe you inherited one that was already mutated. But hey, the good thing is you still have a normal tumor suppressor, okay, and you can still stop cell cycle like you normally did because you still have one good one. So it's kind of like if your car had two brakes, right, and you lost one, but you still had one that worked. The problem is when you get a second hit, and you knock out that other BRCA1 or retinoblastoma or P53 tumor suppressor, now you have no good tumor suppressors for that specific function, right? So now if you are a person that inherited BRCA1 and you gain another mutation in your other BRCA1 allele, now you have a really high probability of getting some kind of cancer, especially breast cancer, because you have no good tumor suppressors that are going to help work in that DNA repair mechanism. So we talked about the two-hit hypothesis with tumor suppressors. Now, oncogenes are a little bit different because once you have one gas pedal stuck on, it can override the other one, right? So once you say go, you're going. Versus once if you have two brake pedals and one goes bad, you can still stop with the other one, right? But if you have two gas pedals and you push on one, doesn't matter if you're not pushing on the other one, it's still going. So with tumor suppressors, we talk about the two-hit hypothesis. And if you inherit one hit already, that's when people have a higher susceptibility or predisposition to cancer. So people with the BRCA1 mutation in one of their alleles doesn't mean they're going to get cancer. It just means you have a higher chance of getting cancer because you only need one more hit. Compared to those of us who inherit two healthy or non-mutated alleles, then you've got to get one hit randomly, and then you'll have to get a second hit. And so you can see the chances of getting two mutations in the two alleles that are on different chromosomes for that same gene is much lower than getting one hit and having one already knocked down. So this is when people are getting BRCA1 screening and trying to decide what to do with that information. And to follow up with that, we have genetic instability kind of for two causes. One, we can get disruption in DNA repair mechanisms. Okay, so this is just showing you all these different things that can mutate your DNA and what they do and our repair mechanisms. Okay, so if you knock out one of these repair mechanisms, you're going to have genetic instability, which just means that you have a higher mutation rate. That means you have a greater chance of accumulating mutations. Sorry. Right? And so it makes you have a greater chance of accumulating mutations in critical genes. Excuse me, or falling into that two-hit hypothesis. So you can have genetic instability due to disruptions in DNA repair. You can have genetic instability due to disruptions in chromosome division. And this is called non-disjunction. And this is when, during meiosis, or it could be during mitosis, during mitosis, or it could be during meiosis, but for you, it matters during mitosis, when your chromosomes don't split properly, right? And so what's going to happen is this cell is going to have 4 chromosomes. This cell is only going to have 2, when they each should normally have 3. And what happens when you have an incorrect number of chromosomes, that's called aneuploidy. So aneuploidy is a result of non-disjunction, and it means you don't have the right number of chromosomes. And so you can imagine how this can cause issues. So this cell will probably just die because it doesn't have all the information it needs. A lot of times this cell will die, or you're going to get maybe excess expression of a proto-oncogene or an oncogene, because now you have 2 copies of it in the cell instead of 1, which really, well, they're not showing even numbers of chromosomes. I should not overthink this. Anyways, the key words here are aneuploidy. When you have the wrong number of chromosomes, it's caused by non-disjunction, and the non-disjunction happens when the spindle fibers don't properly attach. So say this guy didn't attach to his chromosome, and when they split, both of these went to this new cell. I think that's all I wanted to say about that. All right. So there are some additional videos that I'd like you to watch. Primarily the Introduction to Cancer Biology, parts 1 through 4. They're pretty interesting. I have not re-gone through pathways in the chapter objectives. Look through those, have those out for your quizzes. And I made a note, keep your book open to page, that's good English, to the page that has the hallmarks of cancer for your quiz, because there are some questions on that. Again, this is an online class. I expect you to have notes and stuff out, so I don't expect you to memorize all of these. All right, I hoped those helped, and that's it.